Hello and everyone. Welcome to class 12 biology. In this video, we are going to discuss two interesting concepts in genetics. They are polygenic inheritance and pleiotropy. So in this video, at the beginning of the video itself, I want to give you the definition of these two terms. What is polygenic inheritance and what is pleiotropy? So you might want to copy it down in your notebooks. So what is polygenic inheritance? Polygenic inheritance, it occurs when one characteristic is controlled by two or more genes. And the traits which are controlled by many genes are called as polygenic traits. So the word itself is self-explanatory. Poly means many, genic means genes. So a character which is controlled by many genes, it is called as polygenic inheritance, and the trait itself is called as polygenic traits. And the second one is pleiotropy. So what is pleiotropy? Pleiotropy is the exhibition of multiple phenotypic expression by a single gene. When one gene uh, controls multiple phenotypic effects, such occurrence is called as pleiotropy. And the genes which control many phenotypes, they are called as pleiotrophic genes. Right? So pleiotropy means it is exhibition of multiple phenotypic expressions by a single gene. So that is called as pleiotropy. So these are the two important terms that we are going to discuss in detail. I will give you some examples that are provided in your textbook. I will try to elaborate those examples. So let's start. So for Gregor Mendel, we all know that he selected pea plants. And in pea plants, he studied 7 different characters, and for each character there were 2 contrasting traits. So what do we mean by that? Let's look at the example of flower color in the pea plants. In the pea plants, if you look at the flower color as a character, it has got 2 contrasting traits. The pea plant can either produce purple flower or it can produce white flower. So these are the 2 contrasting traits. There were no in-between colors for the pea plants. If you look at plant height, there were either tall pea plants or there were short or dwarf pea plants. There were no medium-sized pea plants. So for Gregor Mendel, it was easy for him. He had 2 contrasting traits for each character, right? And he suggested, or he found out, that for each character there was 1 gene. So there was 1 gene controlling 1 character. So for Gregor Mendel, his selection of pea plant was quite strategic, or maybe he was lucky. So he got all these characters with 2 contrasting traits, and all these characters were controlled by a gene for each. So that was the case. But for other organisms, unlike pea plants, the inheritance pattern and the characters are not as simple as pea plants. So let's take a look at human skin color. We don't have 2 contrasting traits for the human skin color as a character. If you look at human skin color as a character, we cannot have uh, two contrasting traits. Either we have a very light skin colored individual and very dark skin colored individual, and there is nothing in between. It's not like that. In human beings we see continuous variation, right? In human beings we see continuous variation in terms of human skin color. We have got different shades uh, of uh, of human skin color. So let's see over here: very light, right? And then we have medium, and then we have very dark. So the darkness of the skin increases in this particular order, and we see many variations in between. It is called as continuous variation. So same goes for human height as well. Human height, there are people who are short, then there are people who are a little bit taller than the short people, medium height people, and then there are people who are very tall as well. So human height is also showing a continuous variation as a character. There are not just 2 contrasting traits like in Mendel's pea plant. So, uh, so why does human skin color show such variations? Right. Uh, Human skin color, it's an example of polygenic inheritance. Uh, there are many genes which control the human skin color. Right, there are many genes. In fact, there are more than 3, but in our textbook it is given that let's assume there are 3 genes which are controlling human skin color. Right. So let's assume there are 3 genes, gene A, B, and C, which controls the human skin color. The dominant alleles of these genes, A, B, and C, the dominant alleles of these genes are responsible for darker skin color, and the recessive forms of these genes, let's say a small a, small b, and small c, the lowercase a, lowercase b, lowercase c, those genes are responsible for lighter skin color. So A, B, C, I said the capital A, capital B, and capital C, they are responsible for darker skin color because they are involved, these genes are involved in more production of melanin, right? Individuals who have got these genes, right, the dominant forms A, B, and C, right, they are involved in production of melanin, and melanin is the pigment which makes our skin dark. And the lowercase or the recessive forms of these genes, they are involved in less production or no production of melanin in our skin, so that makes our skin light. So these are the genes that are involved in human skin color. So look over here, we have got 3 genes, that is more than 2, right? So we have got 3 genes which control human skin color. So that this is just an assumption we are taking. And in polygenic inheritance, we have to also think about environmental effects of the environmental effects on the phenotypes as well. So now let's try to understand how these 3 genes, gene A, B, and C, which are located on different chromosomes, how do these 3 genes control the human skin color? Let's try to understand that. So in this picture, I have given you 3 individuals, one with very dark skin, one with very light skin, and one with medium skin color. So if we have to guess what will be their genotypes, so it will be easy for these two. The one who is having very dark skin coloration, it is obvious that he will have all 6 of its alleles as dominant alleles. So if all the 6 alleles are dominant, the person will have very dark skin color because he will be the skin cells of this particular person who is having this particular genotype will be synthesizing the maximum amount of melanin pigment, and his skin color will be the darkest. And for this individual, since he is having the lightest skin color, very light skin color, he will not have any allele as a dominant allele. So there will be no dominant allele in this particular person. So therefore, his skin color will be very light. Why? Because there are no dominant alleles. Since there are no dominant alleles in him, he will be not synthesizing as much melanin as the others. So therefore, his skin color will be a light skin color. But what about the medium one? The person who is having medium skin color. So there are many different possibilities, and I will explain you why. So there are these possibilities, right? I don't want to read each and every one of them. Just observe, and in these genotypes, what is the common thing that you observe? The common thing that we observe over here is that each of these possible genotypes, they have got 3 dominant alleles. So the person who is having 3 dominant alleles as in their genotype, that person will be with medium skin color. So it doesn't matter whether 2 A's are dominant or 2 B's are dominant or 2 C's are dominant. It's not necessary, right? The only necessity is over here is among the 6, the person should have 3 dominant alleles. And if the person has got 3 dominant alleles, his skin color will be medium. So what do we understand over here is that the effect of these alleles are quantitative in nature, right? It is additive in nature, right? The number of dominant allele determines the skin shade. Since this person is having 3 dominant alleles, he will have medium skin coloration. If the person was having 4 dominant alleles, his skin color would be a little bit darker than this one. Right. If this person was having only 2 dominant alleles in his genotype, then his skin color will be a little bit lighter. than this particular skin coloration. So that's what the polygenic inheritance suggests. It is quantitative in nature. It is additive in nature. The phenotypic effect of the alleles are additive in nature. Right? The number of dominant allele determines the skin shade. The more number of dominant alleles a person contains, the darker will be his skin color. The less dominant allele means lighter will be his skin color. So to simplify things, we can say that this person is having D6, means 6 dominant alleles. 6 dominant alleles, person will be darkest. This person is having no dominant allele but 6 recessive alleles. So therefore this person will be having very light skin color. And this particular person, it has got 3 dominant alleles and 3 recessive alleles. So his skin color will be medium. Okay, so that is the polygenic inheritance. And over here in this particular table, we can see the range of skin colors over here, from very light to very dark skin coloration. And over here we can see that we have got 3 genes, gene 1, gene 2 and gene 3. And if all the genes, all 3 of genes, they have got dominant alleles, homozygous dominant, right? For all 3 of genes, if they are homozygous dominant, the person will have very dark skin color. And if the person is having all 3 of its genes homozygous recessive, the person will have lighter skin color. And in between, you can just do the counting. This particular person, who is a little bit lighter than the darkest person, he will have only one recessive allele. So it doesn't matter it is over here the first gene is recessive, or the second gene is recessive, or the third gene is recessive. It doesn't matter. The important thing is he should have 5 dominant alleles, right? 5 dominant alleles for the dark alleles and 1 recessive allele. Okay, so likewise you can see the reduction in the number of dominant alleles in this particular order from right to left. You can see this particular person is having the maximum number of dominant alleles, 6 dominant alleles, and from here it gets reduced. So this person will have 5 dominant alleles, this person will have 4 dominant alleles, this person has 3 dominant alleles, and so on. So the skin coloration also changes in this particular manner. So that is about the polygenic inheritance, and you can give the example of human height as well. Okay, so I want to say it again. It is not that the human skin color is controlled only by 3 genes. We are just assuming for our own convenience. There are more than 3 genes, right? So for our convenience, we are assuming that there are 3 genes. So these genes, they control the human skin color. And in your examination also, you can write 3 genes, A, B, and C, which controls the human skin coloration. Now, what about the environmental effect on the phenotypes? The genetic composition itself alone is not responsible for the phenotypes. The environment also plays a significant part. And this we can understand by giving either this example or the human heights example. I already told you that the skin coloration, the human skin coloration is controlled by many genes, so that is why it is a polygenic inheritance. But it's not necessary that gene alone controls the human skin color. It also depends upon the environment. Let's say you have 3 dominant alleles and 3 recessive alleles, but it does not guarantee that you will have a medium skin coloration. If you expose yourself to a severe heat, if you don't get proper diet, so what will happen? Your skin coloration will not be medium. So it will be somewhere towards the dark, because if you expose yourself too much to the sun, you will get sunburn, and there will be more synthesis of melanin in order to protect yourself from the sunburn. So your skin color will be darker. So here the sun is playing a role in your skin coloration, and same goes with the diet for human height. Even though you will have dominant alleles for good height, but if you don't get proper nutrition, then what will happen? You will be having shorter stature. You will have your height will be less. So that is the effects of environment on the phenotypes. So now let's move over to the next concept, that is pleiotropy. Like I told you in the definition part, I told you the exhibition of multiple phenotypic expression by a single gene is called as pleiotropy, and such genes are called as pleiotrophic genes. So let's look over here. We have one gene over here, and this gene is controlling how many characters? 3 characters or 3 features. So one gene which affects multiple characteristics, it is called as pleiotrophic gene, and the phenomenon is called as pleiotropism or pleiotropy. Okay, so what example can we give for pleiotropy? For pleiotropy, we can give the example of phenylketonuria. So you should give the example whenever you're trying to answer a question like what is pleiotropy, give an example. Whether the question asks you for an example or not, you have to give this particular example because it is given in your textbook. So the example for pleiotropy will be phenylketonuria. You might want to remember this particular long term over here. So phenylketonuria. It is nothing but it is a disease. It's a genetic disease. And this genetic disease is caused by a mutation in the gene coding for enzyme phenylalanine hydroxylase. So there is a mutation in a gene which codes for an enzyme called phenylalanine hydroxylase. So what does this phenylalanine hydroxylase do is, it breaks down the amino acid phenylalanine. So this is an amino acid which we get from diets. So this phenylalanine amino acid is broken down into other forms of amino acids like tyrosine, and for this breakdown process, phenylalanine hydroxylase enzyme is required. Right? If there is a mutation in this particular gene, the enzyme phenylalanine hydroxylase will either not be produced, or phenylalanine hydroxylase will be less efficient. So what happens is phenylalanine, the amino acid, is not broken down into tyrosine. So in our body, what happens is there will be buildup of phenylalanine, phenylpyruvic acid and other derivatives. It will start to build up in our body because phenylalanine is not being broken down into tyrosine. Okay, so there will be buildup of these chemicals over here, phenylalanine. So the concentration of phenylalanine will increase in your blood, and there will be less amount of tyrosine in your blood. So what will be the effect of the buildup of phenylalanine and less amount of tyrosine will be, because of this buildup of phenylalanine in your blood, phenylpyruvic acid and other derivatives, these are toxic for brain development. So they hampers the brain development. So what happens is, if you have this particular mutation and your body will lack this particular enzyme, your body will start to build up these chemicals. These chemicals will affect your brain and the person will become mentally retarded. So there will be mental retardation. And since phenylalanine is not being broken down into tyrosine, what happens is, tyrosine is required for the synthesis of melanin, right? So that particular dark pigment. So tyrosine is required for the synthesis of melanin. And since phenylalanine is not broken down into tyrosine, your body will synthesize less melanin. Now, if there will be less production of melanin, what will happen? Your skin color and your hair color will fade away, right? Your hair color will not be normal. Your skin color will not be normal. There will be reduction in hair and skin pigmentation. So you will have light hair and light skin, very fair skin. So these all happens, mental retardation, reduction in hair and skin pigmentation. So these all external characteristics are caused by mutation in just one gene. So what does that suggest? That suggests that this particular gene is responsible for these phenotypic effects. Right? So it's an example of pleiotropy. One gene having many phenotypic effects. Right? Since one gene is having many phenotypic effects, so this is an example of pleiotropy. Okay, so I will give you a second example of pleiotropy. The second example will be pleiotropy in pea plants. You will not see this particular example in the pleiotropy section of your textbook, but it is given in codominance section, right? So you may want to write it down as well. So in pea plants, we observe pleiotropy in this particular manner. Let's say we have a gene which is responsible for producing round seed shape, round and wrinkle seed. The gene which is responsible for seed shape. Okay. This same gene is also responsible for starch synthesis in the seed. The size of the starch granules in the seed is controlled by the same gene which is controlling the seed shape. Okay. So if we have got two dominant alleles and a dominant allele and a recessive allele, what we get is round seed. This is the typical Mendelian inheritance pattern, right? If you have a dominant allele, the seed shape will be round. If we have got homozygous recessive, the seed shape will be wrinkled. That we have already learned in monohybrid cross. Okay, but what these genes do is, the same gene, the gene R, what it does is, apart from controlling the seed shape, it also controls the starch granule size. If an individual is having homozygous dominant genotype, what happens is that particular plant will produce seeds which are having large starch granules, and the one which is having heterozygous, it will have intermediate starch granules, and the one which is having homozygous recessive, it will produce small starch granules. So this same gene, the gene R, it is controlling both the seed shape as well as the starch size in the seed. So therefore, this is an example of pleiotropy. So along with being an example of pleiotropy, this is also an example of codominance. So you will find this thing under codominance section in your textbook. So that is all about pleiotropy, okay, and multiple polygenic inheritance. So in the next video, we will discuss about sex determination, and the sex determination will be in humans, chickens, Drosophila, and honeybees. I hope you read about them. Thank you.